Hello, everyone. Welcome to Pocket Size Bio with Dr. Casco. In today's video, we're going to talk about transcription, which is mRNA synthesis. This is the second part of a three-part module, basically talking about protein synthesis. In the next video, we will be talking about translation. Okay, so let's get to it. Transcription is essentially mRNA synthesis, right? You're going to produce a temporary single-stranded nucleic acid based on a DNA template, which you're going to use to produce a protein, right? And in this case, RNA is basically very similar to DNA in the sense that it's a nucleic acid, with the exception that instead of using thymine, it uses uracil, a chemical cousin of thymine, right, to produce the nucleic acid sequence of that gene of interest that we're going to take a look at. Okay, and it is synthesized in the nucleus. Once it's finished being made, it will leave through nuclear pores out to the ribosome, in which the ribosome would actually manufacture the protein using the mRNA template. Okay, so one benefit of being RNA to do this process is that it's short, it's temporary, right? It's a single strand, so it's easier to maneuver, it's easier to manipulate in these molecular machineries. Okay, what you see here in this little diagram is basically an overview of what transcription is, with a little bit of translation here. If you take a look at this, at these diagrams that are inside the nucleus, is basically the DNA, right? And the DNA will unzip, okay, in a particular area of interest, basically a gene, and an enzyme will come in and catalyze the formation of RNA in one particular side of the DNA, basically that template strand, okay, in which you'll form the mRNA strand. After the mRNA is fully completed, it will basically dislodge and move, basically migrate to the ribosome outside of the nucleus, into the ribosome. Okay, so let's take a look a little bit more in detail on how that works. Okay, like I mentioned before, mRNA synthesis occurs in the nucleus, and its whole process is essentially catalyzed by RNA polymerase. So this is a major enzyme that you need to remember, right? And this RNA polymerase only reads one area of the genes. Remember, the DNA is double-stranded, right? But both strands don't actually code for the protein. Only one strand codes for the protein. And that area that codes for the protein, that side that codes for the protein, sorry, is referred to as the template strand, okay? Or the antisense strand, or the non-coding strand, right? And the other side of that DNA that doesn't code for the protein is referred to as the coding side, because it kind of represents the equivalent resulting mRNA that you're going to create, with the exception of T being replaced with U, right? Thymine being replaced with uracil. Okay, and so what occurs, if you take a look at this diagram, RNA polymerase will come in and bind to that particular area that codes for the DNA, and it will start to kind of read through the DNA strand. And as it's going, it's unzipping the DNA. It's reading that coding area, that template strand, and it's forming the mRNA nucleic acid, right? That RNA nucleic acid. So let's say if you see here, this particular area codes for the gene. You have the RNA polymerase will start to read this DNA template, okay? And as it's going in a 5' to 3' direction, it will start adding nucleotides, right, to the DNA template. So it's moving along, it's writing the DNA, and it's It's accumulating these nucleotides in a particular order, and it's bonding them, right, covalently and forming them, right, until you have, and essentially that whole mRNA strand, right? That process in which the RNA polymerase is riding through and forming the mRNA, right, transcript, what you would call, is basically called elongation. Okay, so the polymerase elongates through the strand, referred to as elongation, right? You have this overall mRNA transcript. Okay, this is another way you can call just the mRNA molecule, right? Sequence there. And after it finishes, it detaches from the DNA strand. And before it goes to the ribosome, it does need some modifications, right? If we take a look closer into how RNA looks like, right, there's some little details we need to take a look at. Okay. So my apologies for the, so we'll just let the little, the little bell ring there. Okay. So let's continue on. The mRNA needs to be processed. So that crude transcript that just finished is actually referred to as pre-mRNA. Okay. But before it's used by the ribosome, it needs to be cleaned up. There's some areas that you really don't need to create a protein. These areas that need to be spliced out or removed out are referred to as introns. Okay. There's certain sections in the mRNA that are actually needed, and there's some that are not needed. Those that are not needed are referred to as introns, and those that actually will code for the particular area, right, for the particular protein, are referred to as exons. Okay, so the cell has machinery to cut them out, right? These particular areas, these introns, that's referred to as splicing. Okay, so using molecular scissors and molecular processes, it removes the introns and then pastes only the necessary sections of the mRNA, right? As you can see here, so you have your pre-mRNA, okay, with these introns that we don't need. After they're removed, the remaining exons get glued together, and you refer to this as your mature mRNA. Okay. If you notice, there's some gray, there's some areas here that are referred to as untranslated regions. These are areas that actually help the mRNA or protect the mRNA as it migrates toward the ribosome, right? You have that poly A tail, and you have the cap that could provide some protection from the from moving right toward the ribosome. Okay, so just a little bit more detail. When you look at the DNA strand, right, there's a coding region that actually will provide you with the introns and exons to create the mRNA. But right before that coding region, there's something referred to as the promoter, right? There's a promoter region, and this area is where the RNA polymerase actually binds into to start reading the gene, right? To start making the mRNA. Now, this is beyond the scope of the WACE program, but there are some regulatory factors that can influence RNA polymerase, sorry, to actually begin to read it, or it gets suppressed, right? There are many factors that can come in and influence whether or not RNA polymerase will work. An example here that you can see are transcription factors that can actually promote RNA polymerase to be read and transcribed, right? But there are other factors that can actually repress or stop RNA polymerase from actually doing its work, right? And this is moreover gene regulation, right? A very interesting topic, but it's beyond the scope of the WACE program. All right. So ultimately, right, you have RNA being synthesized. That is the big idea of transcription, right? Now it moves along to the ribosome to be used for translation. I'll leave you guys with some questions here, right? Try to see if you can answer them, and you can leave your answers in the comments below. Thank you guys for watching.